All right, so we're going to talk about fermentation and anaerobic respiration for the first part of this lecture. There we go. And then the second part, we're going to start talking about photosynthesis, particularly the anatomy of a chloroplast. So as a reminder, study guide is posted, right? And that exam is coming up quicker than you think on October 16th. All right, so let's talk about organisms that we would consider anaerobic organisms. So anaerobic organisms are organisms that don't necessarily need oxygen, right? Because they don't need oxygen, they can use other means to obtain energy. Now remember, aerobic cellular respiration, or the cellular respiration that we've talked about so far, needs oxygen as the final electron acceptor. Anaerobic organisms can use anaerobic respiration or fermentation, which we'll define in a second here. Now, some organisms cannot use oxygen. Some organisms can be a facultative anaerobe, meaning that they can use oxygen, but they can also switch the way they metabolize molecules and survive without oxygen. So they can do both. Now, typically, anaerobic organisms or facultative anaerobes tend to be smaller organisms like bacteria and archaea and some fungi. Even our muscle cells have the ability to go without oxygen for certain periods of time. All right, so we're going to talk about anaerobic and fermentation. Anaerobic respiration and fermentation are different from one another, right? So anaerobic respiration is basically cellular respiration, but uses a molecule other than oxygen as the final electron acceptor. Whereas fermentation produces ATP in the absence of oxygen, but doesn't go through all the steps of cellular respiration. Instead, it just goes through glycolysis and the regeneration of NAD+. All right, so we're going to talk about both of these. Let's start with aerobic respiration. So like I said, aerobic respiration is what we've talked about so far, and that's where we use oxygen as our final electron acceptor. So if I move this video screen up here, there we go. Right, we can see that we have our energy-carrying molecules. It gives away its electrons. The electrons go through the electron transport chain and eventually is captured by oxygen and some of those hydrogens to make water. Oxygen is the final electron acceptor. We need to have oxygen in order to accept those electrons so that more electrons can start going through the electron transport process. All right, so that's aerobic respiration. That's what we've covered so far. Now, anaerobic respiration is really similar, but we're not using oxygen, right? So let me move this screen here. So anaerobic respiration is typically going to be organisms like bacteria and archaea. They're going to go through the same process, glycolysis, breakdown of pyruvate, the citric acid cycle. They're also going to go through oxidative phosphorylation. But instead of using oxygen as the final electron acceptor, they're going to use a different molecule, different electron acceptors. So for example, they might use nitrate as the final acceptor or sulfate. Right? They can also have other electron donors besides glucose. They don't necessarily have to use glucose as their primary energy source. All right, so they're still using the electron transport chain. They're still using ATP synthase. We're still in the mitochondria, specifically in the mitochondrial membrane. But if we look at an example of this here, I'm going to move my screen. We'll move it up to the top here. Right, so anaerobic respiration, in this example, uses nitrate as an example. So NO3 negative as the final electron acceptor. So the bacteria E. coli, right, typically we find this in our intestines, has the ability to use nitrate as its electron acceptor instead of oxygen. Now notice, everything happening here looks pretty much the same. We have NADH donating electrons. They're going through the electron transport chain. We have protons being pumped across the membrane, building up a gradient. We're using ATP synthase to synthesize ATP. The only difference is that the electrons, once they're done with the electron transport chain, will actually be given to nitrate, right? Nitrate instead of oxygen, right? So nitrate accepts those hydrogens accepts the electrons along with a couple of hydrogens. And what happens is that we get nitrate converting into nitrite. All right, so again, it's very, very similar. The only difference is that instead of oxygen accepting the electron, we have a different molecule. Could be a variety of molecules, and we just went through an example where nitrate is an example of one of these. All right, so some organisms can use anaerobic respiration. Some organisms cannot. For some organisms, if there is no oxygen, right, they cannot just revert to anaerobic respiration. Basically, they're limited, right? So some organisms can only use oxygen as the final electron acceptor, and they don't have the ability to use other molecules such as nitrate. So they have to find a way to harvest chemical energy without using oxygen. And we're going to walk through a couple of examples here, but they're going to use the process known as fermentation. Now, fermentation has two steps. It has the first step, which is glycolysis, exactly the same as we've talked about. And then the second step is the regeneration of NAD+. Right? This is a step we haven't talked about yet, but we're going to very shortly. All right, so fermentation. There's two steps: glycolysis, regeneration of NAD+. All right, so fermentation. What's the big deal with fermentation? Why don't all organisms use fermentation? Right? Well, there's a problem. Right? The problem with fermentation is that we get a buildup of NADHs. Right? We get a lot of them. So glycolysis is happening as usual. Right? We're producing 2 ATP. We're producing 2 NADHs, right? The cells are using that ATP for energy, and the NADHs are starting to build up. Now, this is a problem because at high concentrations, NADH, which carries electrons, it's an electron carrier, will start haphazardly donating its electrons to other molecules, and when that happens, it causes free radicals to be formed. And free radicals are highly reactive molecules, highly reactive chemicals that actually have the ability to damage DNA as well as cellular proteins. So we don't want NADH to build up, right? We want to have a way to keep the levels of NADH lower so we don't get the creation of these free radicals. So essentially what we have to do is regenerate NAD+, right? So let's take a look at some examples here. So one way that this can happen is through lactic acid fermentation. Right? So we're going to talk about two types of fermentations. The first being the lactic acid fermentation. This is where pyruvate is going to be reduced to make lactate. Right? So lactate being the end product here. And in the process, we're going to turn NADH back into NAD+. So let's look at the example. We have glucose. We're going through glycolysis. We're producing 2 ATP, 2 NADHs, and 2 pyruvates. Great. Nothing unusual here. We've talked about these steps. But now this pyruvate, instead of being broken down into acetyl-CoA, is going to turn into lactate. Right? Because these organisms, they don't have the ability to continue with the rest of cellular respiration because if oxygen is lacking, they cannot use another molecule to substitute oxygen, right? The only thing they can do is glycolysis. But we're left with pyruvate, and that pyruvate is going to turn into lactate, right? Now, the reason pyruvate is going to change into lactate is because this process of taking pyruvate, and making it into lactate will regenerate our molecule of NAD+. All right, so we've created some NADHs here. All right, we're going to take some of those NADHs because remember, we don't want high levels of them. we're going to oxidize them, right? So we're going to lose electrons from NADH, and when that happens, we get NAD+. Now this NAD+ can be used in glycolysis because NAD+ will then be turned into NADH, which is a product of glycolysis. All right, but again, then we're going to use the NADHs and turn it back into NAD+. So this way, glycolysis can still happen, and we're not getting a buildup of NADHs. All right, excuse me. Now, one of the byproducts here is, well, not the byproduct, the product is lactate. And maybe you've heard of lactate before, right? So if you are an athlete and maybe you've experienced this before, but lactate can often build up into lactic acid in our muscles, right? So our muscle cells have the ability to start using just glycolysis and then form lactate when we're running low on oxygen. So think about it like if you're running a marathon or running long distances, right? So we'll get lactate, and lactate will start to build up, right? That can often cause muscle cramps. Now, once we have enough oxygen, say we finished our race, finished our marathon, we're breathing deeply, taking in that oxygen is restored in our body, then this lactate can actually be converted back to pyruvate or can be used by other tissues in our body, right? So likely you've heard of lactate before. Now, lactate, maybe you haven't heard of it in terms of muscles, but maybe you've heard it in terms of if you have a lactose intolerant, right? That's an enzyme associated with the breakdown of lactate, right? So this bacteria, Streptococcus lactis, and other lactic acid bacteria, we actually harvest their ability to make lactate, right? And we use it to make cheese, all right? So it can be a very useful method as well. We utilize it in our everyday life. All right, there's another type of fermentation that we want to talk about, and this is alcohol fermentation. So this is where instead of pyruvate turning into lactate, now we have pyruvate turning into alcohol. So again, glycolysis is happening in the same manner we've described, nothing unusual there. Pyruvate is being produced, but now pyruvate is going to break down into ethanol, right? So again, we're converting pyruvate into ethanol, and this process allows NADH to be used up and for NAD+ to be regenerated. All right, so pyruvate changing into ethanol, we also get a byproduct where CO2 is being produced. All right, so we're also producing some carbon dioxide in this process. And again, we can harness this type of fermentation, this alcohol fermentation, when we make things like beer. All right, so we can utilize it for our own advantage. All right, so just a review of fermentation here, right? We're breaking down organic molecules to harvest energy, but we don't have the use of oxygen. So instead of going through the full process of cellular respiration, we can only go through glycolysis, but then we have to take an additional step in order to prevent the buildup of NADH. And that additional step can either come in the making of lactate or in the making of ethanol. All right, so fermentation is essentially glycolysis with another step to reduce the amount of NADH. Now, remember, glycolysis allows the body to make some ATP, allows organisms to make ATP, but not a lot. So if we compare that back to aerobic respiration, or cellular respiration in the presence of alcohol in the presence of oxygen, we see that there's a huge difference in the amount of ATP that can be produced. All right, so that was a fermentation and anaerobic respiration. I'm now going to quickly switch gears a little bit. We're going to talk about photosynthesis, and we're just going to introduce the structure of the chloroplast. We'll talk about the rest of photosynthesis when we come back to class on Thursday after the Nobel Conference. All right, so it's important to note that plants use cellular respiration, right? But they get their energy source from the sun, right? So when we're thinking about cellular respiration, our glucose comes from what we eat. But in plants, the glucose that the plants have obtained have come by using the sun, right? So we still have glucose. We're still going through cellular respiration in the same manner, whether we're a plant or an animal, right? But where the glucose is coming from differs. All right, so this is where photosynthesis comes into play. So, photosynthesis is the process of making glucose molecules by harnessing the sunlight, right? So if we think about the interplay of cellular respiration and photosynthesis, we have our glucose molecules. Our glucose molecules can go through cellular respiration, creating ATP. It's also giving off some CO2 as a byproduct. Now, on the opposite side of this cycle is photosynthesis. So during photosynthesis, we're using that CO2, we're using that light from the sunlight to produce more glucose. And then again, that glucose can go through cellular respiration, produce CO2. That can go through photosynthesis, produce glucose. It's a cycle. All right, so if we look at the overview of photosynthesis here, we're essentially capturing the energy within light in order to make glucose molecules. Right, so if we're looking at this equation, we see CO2 becomes reduced and water becomes oxidized. Now this whole process has a change in Gibbs free energy of positive 685. That is not energetically favorable. This is not going to be a spontaneous reaction. This reaction requires an input of energy. And the energy in this case is coming from energy harvested from sunlight. All right, so energy from the light drives this endergonic reaction. All right, so what type of organisms can harvest light energy? Well, there are different types of what we call tropic levels. So a heterotroph has to eat food in order to obtain organic molecules. We are a heterotroph. Other animals are heterotrophs. Other insects are heterotrophs. But there's also this class of animals, this class of organisms that we could call autotrophs. Now, autotrophs have the ability to make their own organic molecule. And specifically, if you're an autotroph that uses sunlight as your source of energy, you would be a photoautotroph. So typically we think of plants, right? Plants photosynthesize. But there's also algae, there's also some cyanobacteria, there's also some protists that have the ability to photosynthesize as well. So the ability to harvest light energy. We're going to talk about this in terms of, mostly plants. And so in order to understand how plants use photosynthesis, we have to understand some basic leaf anatomy. Where is this all occurring? All right, so here we have a cross-section of a leaf. If I took a leaf and kind of cut it across and then looked into the leaf, this is an example of what I would be seeing. I would have this top layer of cells. I would have this middle section of cells, and then some other middle section of cells, and then I would have another lower level of cells. Well, photosynthesis occurs in this middle part here. Specifically, it occurs in the mesophyll cells. Right, so these mesophyll cells are the cells inside of the leaf. There are two different types, right? And you'll notice that they're inside of the cell, there are two areas that look a little different. We have the palisade mesophills, which are long and columnar shaped. And then we have the spongy mesophyll cells, which are round, a little bit more irregular, there's more open spaces. All right, so photosynthesis is happening in these mesophyll cells. Now, in order for photosynthesis to occur, we have to get carbon dioxide into the cells, into these leaf cells. And that is done by carbon dioxide entering in through these openings on the surface of the leaf called stomata. All right, now the opening itself is called a stomata. The cells that create the opening have a different name entirely. These are actually called guard cells. So it's just the pore, it's just this opening that we would call the stomata. This is where CO2 is allowed to enter into the leaf so that it can be used during photosynthesis. We'll also see that during photosynthesis, oxygen will be created. So oxygen can leave through these same stomata. And plants have the ability to transport water. Water can also leave, can evaporate through these stomata as well. So we have a lot of gas exchange occurring at these stomata. All right, so we're in the mesophyll cells. That is where photosynthesis is occurring. Right? And the stomata are the openings in which we can get the CO2 in order for photosynthesis to occur. All right, so we're in the mesophyll cells. Excuse me. Specifically, we're looking at the chloroplasts, which is the organelle in the mesophyll cells responsible for carrying out photosynthesis. Right, so these mesophyll cells are going to have a lot of chloroplasts. In the chloroplasts, we're going to find a pigment molecule called chlorophyll. And chlorophyll is going to be responsible for harvesting some of this light energy. All right, so we are in the spongy mesophyll cells. Specifically, we're looking in the plant cell, right? And as we're looking in a singular cell here, we are specifically focusing on the organelle, that is the chloroplast. And within the chloroplast, we have pigments that are called chlorophylls, right? So they're actually embedded within the chloroplast themselves, these pigments called chlorophyll. And we'll see how chlorophyll plays a role when we talk about the process of photosynthesis. All right, so let's look at the chloroplast anatomy. So a chloroplast anatomy, it's one of those organelles that are semi-autonomous. Remember, they can divide and grow on their own. They have their own set of DNA. And a chloroplast contain a lot of membranes, right? They contain a ton of membranes. So first of all, we have two membranes surrounding the organelle. We have an outer membrane, we have an inner membrane. And then once you go inside of those two membranes, we have this network of other membranes kind of squished together inside of this thylakoid, or inside of this chloroplast. And these membranes that are all kind of compacted together are called thylakoids. Right? So these are thylakoid membranes. And within the membranes themselves, this is where we will find the chloroplast pigment. Now, one of these kind of like pancake-looking structures would be a thylakoid. If we put a lot of these thylakoids together into a stack, this is called a granum. If we look at the space inside of the thylakoids, right, this space inside of the thylakoid, that is a fluid-filled region inside of the thylakoid. That is called the lumen. And then the region outside of the thylakoid membranes is called the stroma. Right? These terms will be important when we start talking about the process of photosynthesis. All right, that's all I had for you. I hope you enjoy Nobel or have enjoyed Nobel. I will see you on Thursday.